Hello everyone, my name is AJ Keefe, and in this presentation I'll be covering the paper, A Molecular Signature for Anastasis, Recovery from the Brink of Apoptotic Cell Death, published in the Journal of Cell Biology. I want to extend a special thanks to the authors for their contribution to this field of research, and I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before playing my commentary. If you have any questions, please comment below, and I hope you enjoyed this presentation. This process of anastasis is actually a very recently coined and recognized process whereby a cell that appears to have entered the terminal stages of apoptosis or cell death will occasionally recover. So the final stages of apoptosis are marked by widespread caspase activation, including, most importantly, caspase 3, which very potently induces membrane blebbing and the formation of apoptotic bodies for phagocytosis. So caspase 3 also cleaves a protein called ICAD, ICAD, or the inhibitor of caspase-activated DNase. So ICAD stands for caspase-activated DNase. And this, when caspase 3 cleaves it, it unleashes CAD, to begin mass cleaving and digesting DNA. Caspase 3 also cleaves an inhibitory domain of a protein called gelsolin that functions to disassemble and cleave actin. So when caspase 3 cleaves gelsolin, this actin severing protein becomes constitutively active and it just begins destroying and leveling the cytoskeleton of the cell. And this leads to membrane blebbing. So caspase 3 activation is basically synonymous with DNA cleavage and cytoskeletal destruction and many other deleterious events throughout the entire cell. So the big question is when or how does a cell occasionally survive this complete catastrophe of caspase 3 activation? How does a cell expressing activated caspase 3 recover? And when they do recover, which sometimes they do, what signaling events or transcription profiles mediate that process of cellular recovery? And so we're going to be looking at this process of anastasis. It's basically recovery from the terminal stages of apoptosis. And so before answering these big questions, we need to also cover this process of an EMT. And it's occasionally associated with, or usually associated with TGF-beta signaling. This is the TGF-beta signaling pathway that we're going to get into in a second. So the EMT stands for epithelial mesenchymal transition, and that's what it is. It's basically when an epithelial cell reprograms itself to function as a motile mesenchymal cell, like a fibroblast. So this EMT is basically so just like imagine a rigid column of epithelial cells turning into these squishy motile fibroblasts, and this is what the EMT essentially is. And fibroblasts are very interesting cells because they are a pluripotent cell. They're not totipotent, so they can't turn into any cell, but they're pluripotent, so they're limited to certain cells that they're able to form, but they are dedifferentiated compared to the epithelial cells. So a shift from epithelial cells to a mesenchymal-looking fibroblast is marked by the gain of a trait called stemness, or the ability to differentiate into other cells. Essentially, it looks more like a stem cell than it did before. So the EMT essentially turns these differentiated epithelial cells into more stem cell-like fibroblast cells. So the question is, how does and why does an epithelial cell turn the clock backwards to form these motile mesenchymal cells? So the EMT actually happens during wound repair, and that's the canonical event that activates the EMT is during injury, during cuts or bruises. So this kind of makes sense too, because when an epithelial lining is damaged from something like a cut, the epithelial cells begin proliferating and they fill in the lost tissue. And then they physically move into the wound to fill it back up with cells. So the EMT allows epithelial cells to begin moving, but it also allows epithelial cells to detach from the extracellular matrix. And so in order to become motile, obviously an epithelial cell that dedifferentiates needs to get rid of some of its attachments to the extracellular matrix. And this is done primarily through the loss of E-cadherin. Right through down here, we can see E-cadherin production is halted. This forms the attachment to the extracellular matrix, or sorry, E-cadherin, it forms attachment with adjacent cells. So normally when a cell loses its attachment to the extracellular matrix or to other cells, it actually enters a process called anoikis. So the ability to survive without integrin attachment to the extracellular matrix or with E-cadherin attachment to neighboring cells is actually a very big privilege for any given cell. And only in very rare conditions, such as during wound repair or during an EMT, are cells actually allowed to withdraw their extracellular connections. And this is generally a mechanism to prevent carcinogenesis and metastasis, because the process of metastasis is obviously it requires a cell to detach from its matrix and physically tunnels through tissue and enters the bloodstream. And normally when an epithelial cell dies very quickly, when it loses its attachments to the extracellular matrix. And again, this is mediated through a process called anoikis. Anoikis is a type of programmed cell death caused by the loss of integrin attachment. So the integrin attachment to the extracellular matrix actually produces pro-survival signaling. And when this is lost, the epithelial cell will commit suicide through anoikis. It'll self-destruct. However, in EMT, this EMT is essentially a transcriptional event where it allows a cell to survive without extracellular attachments, and so it inhibits this anoikis event, and it basically promotes gene expression associated with fibroblasts and allows motility. So how is an EMT initiated? Well, an EMT is initiated through a combination of signaling pathways. Most notably would be some of the really important ones would be WNT or WNT, notch and patched and smoothened signaling. These are all signaling pathways. But most critically for the EMT is this TGF-beta pathway, TGF-beta. So this TGF-beta is a signaling protein that is normally bound up in the extracellular matrix in an inactive form, but it can be proteolytically cleaved or sometimes released during injury-associated inflammation. And when TGF-beta is released from the extracellular matrix, it can bind to a receptor on epithelial cells, and it initiates this SMAD signaling pathway. So this TGF-beta receptor is a serine threonine kinase that, when bound to TGF-beta right here, it will oligomerize and begin phosphorylating intracellular proteins. The primary target of the TGF-beta receptor are of the SMAD class, so these SMAD proteins. When SMAD2 and 3 get phosphorylated, they translocate to the nucleus, along with this super important co-SMAD that allows nuclear translocation. And when they get into the nucleus, they activate gene expression. And important for this paper is snail. We see snail right here. So this snail transcription factor is the number one most critical transcription factor for inducing the EMT. And without snail, although the EMT is still possible, it's largely inhibited without snail. And snail, which is activated by the SMAD transcription factors, begins initiating a second round of transcription that is required for the mesenchymal phenotype, which includes things like motility, proliferation, and various survival factors. So just to summarize, we have anastasis, which is recovery from the terminal stages of apoptosis, marked by caspase-3 activation, right? And we also have this interesting EMT phenomenon that's marked by TGF-beta induced SMAD signaling and a subsequent upregulation of the snail transcription factor. And the snail transcription factor that was activated by the TGF-beta signaling then upregulates mesenchymal gene expression profiles. So the big question is, why am I talking about the EMT and anastasis in this slide? And we're going to see in this paper that the EMT and recovery from cell death are very similar processes that have a lot in common and a lot to do with each other. The current paper is simply asking the question, what signaling pathways are responsible for anastasis, for the recovery from the brink of death? How does the cell climb back out and survive caspase-3 activation during apoptosis? So to answer these questions in a robust, unbiased approach, they began with a whole transcriptomic RNA sequencing of cells recovering from apoptosis. And they wanted to see what genes were most upregulated during these periods, these initial periods of recovery. So they induced apoptosis with ETOH, which induces DNA damage, or sometimes they use STS, a kinase inhibitor, and they observed robust caspase-3 activation, so they see it everywhere, and then they sequenced their RNA profiles, and they found, following recovery from widespread caspase-3 activation, they found two unique sets or stages of transcription following apoptosis. The first few hours of apoptosis were marked by significant TGF-beta signaling and SMAD phosphorylation, and the later second stage of recovery was marked by snail expression and EMT induction. So they found that inhibiting the entrance into the EMT essentially blocked cells from recovering. So in other words, recovering from the brink of death hinges on TGF-beta induced snail expression. So they also noted that autophagy mediated anastasis, so recovering from severe starvation marked by upregulation of autophagy, was a distinctly different process than recovery from severe apoptosis. And so that's an interesting facet to this paper, and that suggests that there are unique pathways and mechanisms of anastasis. But for apoptosis, the apoptosis they were largely studying in HeLa cells, and they also looked briefly at H4 neuroglioma cells as well. But they found in both these cell types that the TGF-beta SMAD snail signaling pathway was absolutely critical for survival following apoptosis. And so this paper has huge implications in general for probably most notably cancer, because cancer cells tend to initiate the EMT following something like chemotherapy. And so presumably the ability to block the EMT during chemotherapy when those cells are at the brink of death could promote or could block the cancer cells from recovering. And also in a more general sense, understanding how cells survive severe apoptotic conditions can also have major implications for many diseases like Parkinson's disease or Huntington's disease or diabetes or you name it. So the recovery from cell death mediated by TGF-beta, SMAD, and snail. Let's get into the specifics. The first step to tackling these big questions is to simply take some cells, like HeLa cells, and induce apoptosis, and then see what genes are being transcribed during the process of recovery. So in panel B right here, we see 3 hours after ETOH treatment, and we see the classic characteristics of apoptosis. We see membrane blebbing, rounded shape, and we see gradual recovery by 4 hours. So by 4 hours, they're beginning to look normal. And to confirm that apoptosis was being adequately induced, they also checked PARP, or PARP cleavage, which is a well-established substrate of caspase-3. So the amount of cleaved PARP is an assay for amount of caspase-3 activity in a cell. When they see that when ETOH is T, which is treated, they see robust cleaved PARP. So when ETOH is administered, we see caspase-3 activation. So we know these cells are entering apoptosis. Then they took these cells at different time points and they sequenced their mRNA. And in this heat map, we see two distinct stages of recovery. We see an early response set of genes. So these genes are classified as the early response. They occur roughly 1 to 4 hours after or following the pulse of ETOH and the recovery. And then we see another distinct set of transcription occurring around 8 to 12 hours. So we have two sets of transcription. And now let's examine exactly what those genes are that are being upregulated. In the early response genes, we see most critically the upregulation of TGF-beta signaling pathway genes that induces, so this TGF-beta signaling pathway, again, is going to be upregulating snail and is associated with SMAD phosphorylation. We also see W&T, Wnt signaling pathway, which is also associated with the EMT induction. And in the late stage, in the later response genes, we see things related to protein homeostasis, like RNA transport, protein processing in the ER, and spliceosome and ribosomal biogenesis. So these late response genes are related to basically recovery and the cells trying to return to a normal state. But the authors looked at this and they saw that the TGF-beta signaling pathway being way upregulated, and they wondered if just how important this pathway was to recovery, and they wondered if it was inducing an EMT. To see if the cells were entering an EMT, they looked at two basic properties. They were looking at cell proliferation. So here we're seeing the cell proliferation and motility. So they found that anesthetic cells, as opposed to the control cells, were far less proliferative. So these cells that were injured and recovering were not proliferating as quickly, but they were moving much more quickly. So in this figure to the right, we see that by 13 hours, the anesthetic recovering cells had almost completely filled in this wound. So they induced a little wound by just cutting it, and they had moved in by 13 hours, while the control mock treated cells had not moved in. So they're actually moving more quickly after recovery from apoptosis. So the authors wondered if anastasis induced by apoptosis has anything to do with anastasis induced by autophagy. So they took various genes that were upregulated during recovery from apoptosis induction, such as snail, obviously, or all these other genes. We also looked at the recovery from autophagy, and they found that many of the genes that were upregulated during apoptotic recovery are not upregulated in recovery from autophagy. And we also don't see cleaved PARP, so we know the autophagy pathway is distinct from apoptosis. There's no apoptotic induction during starvation, and that's to be expected. So what this basically suggests is that autophagy-mediated recovery from cell stress is distinctly different from apoptotic recovery. So there may be multiple mechanisms of anastatic recovery from the brink of cell death, whether it's induced by caspase-3 activation or whether it's induced by severe starvation. There may be multiple pathways by which a cell survives and recovers. Since cells recovering from apoptosis appear to be upregulating various proteins related to the EMT, like snail, and they also show the characteristics of an EMT, like motility, the authors wondered if the EMT was critical for anastasis and recovery from apoptosis in general. Indeed, they found that inhibiting snail following ETOH or STS induced apoptosis in a significant number of recovering cells. So in these first two bars, let's start with these two bars right here. They have scrambled shRNA control, and we see that STS treatment induces recovery in about 50% of cells. This is under normal conditions. When you add snail shRNA, we see 100% of cells are surviving normally. Not surprising. There's no apoptotic induction right here. But when you induce apoptosis, only roughly, 5 or 10% of cells survive. So the recovery rate drops from 50% to 5 to 10% recovery rate. So the presence of snail is very critical to the process of recovery in anastatic cells. We see a similar phenomenon between ETOH-induced apoptosis. It drops from around 60% to around 40%, and it's significant to a very high degree. In the top right panel, we see that inhibiting TGF-beta receptor using a small molecule, this Li36447, this inhibits the TGF-beta receptor. And we see that when you inhibit the TGF-beta receptor, so these two bars to the right, these are the interesting bars. We see ETOH is present in these two bars. We see the recovery rate, or the expression of snail, the expression of snail drops to around 10% from 30% relative to the mock control. So in other words, TGF-beta receptor is critical for the expression of snail. In this bottom left panel, we see a Western blot confirming the presence of phosphorylated SMAD. So we see phosphorylated SMAD spikes in ETOH-treated cells following recovery. We don't see it present in starvation. So that's perhaps why starvation mediated anastasis is not associated with snail or TGF-beta, because there's no phosphorylated SMAD here. We do see phosphorylated SMAD over here. We also see the upregulation of snail. So this collectively suggests that TGF-beta receptor mediates SMAD phosphorylation and the subsequent snail upregulation. And the loss of snail is very detrimental to recovering cells, as we saw in this panel. Thus, TGF-beta SMAD snail signaling, that pathway, is critical for the survival of cells following apoptosis induction. So the summary for this paper is that anastatic cells at the brink of apoptotic cell death are able to occasionally recover by upregulating the TGF-beta SMAD snail signaling pathway and initiating the EMT program. Additionally, anastatic gene expression induced by apoptosis is distinctly different from anastasis associated with starvation and autophagy, suggesting a unique mechanism of recovery. The study reveals that TGF-beta induced SMAD phosphorylation and subsequent snail upregulation is a critical survival event in the recovery from apoptosis. So I hope you guys enjoyed this paper. If you have any questions or comments, please post them. And thanks for watching.